Hi, my name is Daniel Martinez, and I'm joined here today by my colleague Tomas Friman. We represent Pelago Bioscience, a contract research organization with a quest to deliver biologically relevant target engagement to anyone working in drug discovery. And in our talk today, we wanted to introduce you to the cellular thermal shift assay, and we want to show how we use this method to build a large database of cellular response patterns to different drugs and probes. We also want to share with you some examples from the database where the choice of cell system proves to be extra important. The reason we are here today in this specific TMT virtual event is that we rely heavily on the use of TMT-labeled peptides for our work, both towards customers, but also in our internal R&D work. We are grateful to Thermo Fisher for giving us this opportunity to share our data. I want to start by going through the working principles of this method that's so dear to our heart here at Pelago. The cellular thermal shift assay, or CETSA in short, was invented in 2011 in the labs of Professor Per Nordlund. In 2013, we published the very first paper on CETSA, and in this paper we proved the concept of the method, which was that thermal shifts can be measured in the intact cellular environment. You can see the principle in the cartoon here on the left, where a typical protein melt curve in blue and the typical melt curve of the same protein in a more stable form as it has bound to a compound, a ligand, for example, in dashed pink color. This cartoon shows what any biophysics textbook had already been showing for decades. So thermal shifts, they weren't new in 2013. They had been used extensively to prove, for example, ligand binding to proteins or to identify optimal buffer conditions to obtain a stable protein. But until 2011, this had all been performed on purified proteins in isolation. The new thing here, and what had such inventive height that we were able to patent the method, was that these shifts, instability, they also occur in the cell, and we had found a way to study that. In this paper, we showed how well-known drugs on or off the market had a stabilizing effect on specific proteins, their target proteins in the cell. You see some of those examples in the screenshot on the right. One of the important aspects was also that the method required no tampering with the proteins. There are no tags needed, and the compounds that you study don't have to be labeled or in any other way modified. A biologically relevant system, in other words. This paper and the technique was very positively received. However, the data was entirely based on Western blots, and therefore we studied only compounds where the target protein was already known, and we had working antibodies for it. We had been playing around a bit with mass spec detection during the development phase, but it wasn't until the paper was published and Misha Savitsky and his colleagues at GSK CellZone saw the paper that the true power of mass spectrometry and CETSA was combined for the first time. The CellZone group were at the time busy working with the then new TMT10 format and immediately saw a nice fit for the two and the potential of not just looking at the melt and shift behaviors of the anticipated target, but also to identify off targets in more of a discovery mode. We teamed up with them, and in 2014 we published this article here together, where we described compound specificity, mode of action. We explained some unwanted side effects from certain drugs. I should also say that the protocol has seen lots of incremental improvements during the years, which is why you may have heard of CETSA under different names. For example, as here it's referred to as thermal protein profiling, or TPP, but we also have PISA, we have CETSA, and the latest addition that I know of is PIZA from the Haigis and Gigi labs. Following these two science papers from 2013 and 14, we have seen a fantastic uptake in the academic world. CETSA is nowadays not only used to prove early target engagement, which is the binding of a molecule to a protein, but it's also used to describe the initial molecular events of a cellular response, and then linked with alternative cellular assays to help explain later stage cellular processes or phenotypic results, resistance development, or just to name a few. The pizza assay that I mentioned is one such example where phosphoproteomics was linked to cellular thermal stability mapping and used to discriminate between cellular states. I'm not aware if this work has been published, but it was presented at the ASMS meeting in June this year. We also see a great use of CETSA in the drug discovery industry. The uptake has been phenomenal, but here instead the focus is usually on better understanding the working mechanisms of a particular drug or drug candidate. And we at Pelago Bioscience, we have been active for 10 years now, today serve a customer base of more than 100 companies, among them biotechs, small and medium, as well as at least the top 10 largest pharma, both providing services but also offering licenses to the method. We're most active in the early hit ID and hit to lead, and then on to lead opt. And in some cases, we also have the pleasure of translating assays that we develop for our customers early on to follow them into preclinical development in various model animals and even to the clinic. I will only touch briefly on this one, but I wanted to say that CETSA assay comes in different fit-for-purpose formats. We have the Navigate format up there, which is a targeted format. We either use antibodies in protein-specific assays. Here we either work with the late-stage projects to prove target engagement and connect it to efficacy, or we start really early when a target has been identified and screen large libraries of compounds in a plate-based fashion. We either screen compound libraries ourselves here at Pelago, or we export them, most often to our licensees, so that they can screen in-house. Recently, we have also developed a targeted MS protocol to allow us to work with proteins where no antibodies are available yet, or where we want to multiplex, follow a number of proteins in panels at the same time, but not necessarily go for the full MS readout. Instead, we also offer the CETSA Explore format down there, which is where we focus on during the remainder of the talk today. This is a protein-wide MS-based detection format enabling unbiased compound profiling. It's useful for target deconvolution following, for example, a phenotypic hit, selectivity studies, and to understand the mode of action of your compounds. Starting off by showing what results normally look like in our hands. You can see the melt curves on the left here. Show how the protein ERK is gradually stabilized by the addition of more and more of the compound ravoxertinib. The blue line shows the melting of the naked protein, and then increasing concentrations follow to the right. And these melt curves, they stem from a plate-based assay using alpha screen, but the heat map on the right is the same compound in CETSA MS in the so-called 2D format, showing you basically the same behavior as the curve on the left, but with increasing shifts in blue intensity there. In order to generate this kind of heat map, we needed at least 50 data points just for an N of 1. And this, to no one's surprise, means extensive machine time and reagents costs. So we needed to do something about that. We used this format for a few years, but realized that we could do a one-pot approach instead, where we pool all the temperatures into one sample. We had done this already back in 2011 when we had just started developing the method. The scale of experiment was much smaller then, and the space on gels were scarce. So in principle, we collect all temperatures at every added concentration of compound, and then plot the area under the curve for each concentration as an amplitude compared to the control sample. The data can then be visualized in different ways, but we normally go for volcano plots, like the one on the right here, where we have plotted just the highest concentration amplitude in the green box. And this particular example shows how methotrexate binds to DHFR, a high confidence hit up there. But you can also see several other proteins involved in the folate and thymidine metabolism show up. We call this format compressed CETSA, but it's also known as PISA or one-pot CETSA. Now, when we use MS readout, where we normally look at the melting behaviors of some 8,000 proteins in one go, we're not just looking at proteins that shift in stability as a consequence of binding the added compound. We're observing how protein interaction patterns change in the cell. So we're looking at both chemical binding to some proteins and then stability shifts from biology-driven effects, where one protein may, for example, be hypophosphorylated as a secondary consequence of compound addition. And this may be picked up in our data because a shift in phosphorylation state could affect interaction patterns with other proteins in the cell, and that also affected thermostability of that protein. This plot shows you that there are indeed those proteins that are destabilized by addition of compound. And we find that destabilized proteins are usually those that are deprived of a binding partner as a secondary consequence of compound binding. For example, that same hypophosphorylated protein again, or it could be a protein that shuttles from one organelle to another where conditions are different. And there could also be purely chemical explanations where the compound binds to a partially unfolded protein and more influence the solubility of the protein in a fashion that is not connected to its pharmacological effects. Anyways, the compressed format allows us to more easily assess the data since we don't have to fit melt curves to all proteins. The sample handling is easier, and we can also use the TMT channels to gain statistical power by more replicates. I wanted to show one example of how this method format has been used. So in this article from 2022, this group investigated the compound allantoactone. It's a well-known compound in Chinese medicine. And up until this publication, the target for allantoactone was unknown, but the authors used CETSA compressed protocol and identified aldoketo reductase family C1, so AKR1C1, as the target. However, the proteomic depth was only 2,400 proteins, and the data quality did not allow the authors to identify more potential target proteins. We see large shifts here in the volcano plots, but statistics-wise, they didn't really make the cut. One potential explanation is that they incubated with the compound for 2 hours, which could have effect on the cell that are not only due to changes in the interaction patterns. In follow-up experiments, they show that the drug has an effect on recombinant AKR1C1 activity, much more so than for the protein subgroups 1C2 and 1C3, but still at 10 micromolars, the compound seem to have a weak inhibitory effect also on 1C2 and 1C3. When we applied our protocol, which in theory is the same, but with 1 hour incubation, very stringent sample workups, we reach a proteomic depth of a bit more than 8,000 proteins, and we see excellent statistics. We do pick up the same target protein, but also other family members, as you can see in the volcano plot. But there are also a number of proteins where the data shows both stabilizations as well as destabilizations, and they follow clear dose responses like the one on the right here. It also looks like the compound is equipotent on the 3 family members, but there are also other proteins that respond in the same concentration range, perhaps as a downstream events. I'm going to make a pretty big number in the following slides about the importance of picking the right cellular system. We will show examples of where the choice of cell line is important. So therefore, I should probably stress that we performed our experiments here in another cell line compared to the published study. So some of the differences could be attributed to that. But my main point is on the importance of data quality. It's super important to optimize your sample workup and data collections to obtain precise protein quantification. If you don't, it's not going to help that you have a great tool and reagent such as the TMTs. As you noticed from the last couple of slides, CETSA with MS-based detection generates a kind of molecular phenotype, a fingerprint of the cell and how it reacts to the added compound. Again, the image we get is composed of both cellular direct effects from binding and the downstream events. It's not always easy to say what is what, and sometimes we're helped to lay the puzzle from the scientific literature or different databases, but that's not always the case. So in 2018, we started collecting molecular fingerprints in our own database, and we call this the Target Engagement Atlas. The idea was to have a database that we can use to compare the profiles of our customers' compounds with those of well-annotated, commercially available compounds. So if we can't explain exactly what happens and why, then at least we can compare it to known compound matter. The database today consists of about 500 compounds, and many of them have been profiled in several cell lines. The experimental layout has included as many steps as possible to be handled by robotics. So that's fairly straightforward with this compressed protocol. And all data here exists as biological triplicates, and we have both negative and positive controls included in our TMTs. And in total, we spend about 6 hours of machine time per compound. This slide is getting a bit old, but it still serves the purpose of summarizing the results from the first years and the principle of what we can do with our data. It shows t-SNP plot of the first 190-something compounds that we ran in HepG2 cells. And this t-SNP plot is an unsupervised nonlinear reduction model that makes it much easier to visualize higher dimensional data. So in principle, compounds that lie close to each other, they have a similar CETSA profile, or the fingerprint, if you wish. And the further away they are from each other, the less similar they are. So for example, up here, you have mTOR inhibitors. They stack nicely together because they act similarly on the cell. You have kinase inhibitors on the left, but you also see kinase inhibitors scattered throughout the whole plot as they often hit clusters of kinases or have specific and different cellular downstream reactions. Down here you have tubulin inhibitors, and you can see the Vinca alkaloids there, vincristine, vinblastine, vinorelbine, but there is also colchicine among them, and this is because colchicine is an alkaloid. Docetaxel is another well-known tubulin stabilizer. And finally, on the right here, you have an antibiotics cluster in orange, where a single antiviral in green, there, altegravir, shows in a quite interesting similarity to the antibiotics. If we highlight the N-side group of compounds that we included, we see them stack into 3 main clusters. And if we want, we can then zoom in on the data for these specific compounds, compare them to each other to see if the fingerprints that drive the clustering can be correlated to cellular or clinical outcome. So we can look at, for example, diclofenac and lumiracoxib that almost stack on top of each other in the plot here, or piroxicam, meloxicam, and droxicam that also must have similar effects on the cell. And if we zoom in on the proteins that we can see the proteins that drive the clustering. We see up on top here that diclofenac and lumiracoxib, they share this cluster of proteins in the upper left corner that respond to the treatment, and you see the proteins there on the x-axis. And the same is there for droxy, piro, and meloxicam. They share similarity in another region, which makes them cluster together. Also, importantly, sometimes we don't find the primary target in our MS acquisitions, but instead we see the effects on the cell, a bit like ripples on the surface when you throw stones in the water. And this is what I meant about seeing both chemistry-driven binding profiles and biology-driven cellular responses. We also have compounds with different primary targets inducing the same overall cellular patterns. So there are typical fingerprints, for example, for different paths to apoptosis, for example, to redox stress, or to calcium release. And this kind of database is what we're compiling. The results I've shown you so far have been from one single cell line, HepG2, which is a hepatocellular carcinoma, but we have also expanded the atlas to other cell lines, so in K562, which is a myeloid leukemia, and U87MG, which is a primary glioblastoma cell line. And this way, we have a fairly broad repertoire of cell type backgrounds. We were curious how these differences in biology would translate to target engagement profiles and the subsequent responses in different cellular backgrounds. And again, we're still just looking at 1 hour incubations. So the cellular responses that I'm referring to, they are probably just the springboard towards a less well resolved, but sometimes very defined phenotypic effect. What we see so far in this part of the project, and I'm going to be quite unspecific here, simply because we haven't gotten that far in the analysis. What we see is that among the compounds we've assayed in all the cell lines, that's about 100, is that in general they overlap quite well for the primary target if it's there. But then there are a lot of other things happening as well that can be very cell line specific. So this slide is quite busy, but it's a good summary of the data. And you see a large number of compounds where the proteome behaves similarly, sometimes close to identical, but there are examples where this is not the case. The most obvious ones are those that cluster around or along either the X or Y axis in the plots here. For example, you can see that this protein here, ZBTB21, is shifted to the same extent in K562 and HepG2s by procarbazine. But then, comparing U87, there is no response in U87. The same goes for superoxide dismutase 2, which is affected by both lumiracoxib and ruxolitinib treatment here in K562, but not in U87MG nor HepG2s. In fact, there are quite a few protein compound combinations that only light up in K562, whereas not in the other two cell lines. Similarly, we also see that iniparib has several different effects in K562 compared to the other cell lines. And I'm going to finish off with a specific example. The SARC family of kinases. This family is composed of SARC itself, but also YES1, FIN, LIN, and a few more. And here is one example where we believe CETSA can pick up specific cellular protein activation states that makes the protein respond and shift differently between cell lines. And this is obviously super important information to have when you either plan an experiment. You do not want to run your experiments in a cell line that is not going to be responsive, that doesn't represent biology, or the other way around. These activation states could also offer an explanation to unwanted side effects in specific cell types. Okay, so this is another busy slide. I hope I can walk you through it. What we're looking at here is the correlation between how different compounds, so those are the dots in the graph, affect either SARC itself on the y-axis or the SARC family member YES1 along the x-axis. And you're looking at log two-fold changes here. That's the scale of the axis. The different color of the dots denote the cell type. So you should have 3 dots per compound in the plot. And you can see by the line fit here that there is a correlation. It's not super strong between the responses in HepG2, those are the red dots, between these two proteins. And for U87MG in green, there is some correlation as well, but definitely not for the blue one, K562. This indicates that the responses of these two proteins have less in common in K562 than in the other ones. And this is also obvious from the patterns. You can see for the red and green dots that both these proteins are destabilized by the addition of all compounds except ponatinib and neratinib in HepG2 on the right there. And this destabilization is pretty much what we expected from our past experience. Usually when SARC family proteins are treated with compound, they become destabilized. Therefore, we were a bit surprised when one of the SARC proteins, SARC itself, turned out to be stabilized or not shifted at all for many of the compounds that caused clear destabilizations in U87 MGs and HepG2. You can see it there encircled. For ruxolitinib, avapritinib, vandetanib, crizotinib, for example. But we also found that those compounds that do cause a destabilization of SARC in K562, they do so, but to a much lesser extent than in HepG2s or U87 MGs, which can be visualized like this with the triangle here, where the blue dots for K562, they end up near the SARC 0 line, while having a strong destabilizing effect on JES1. These triangles, they also highlight that JES1 is affected to a lesser extent by nintedanib and ripretinib in U87 MGs in green there on the right than in K562 and HepGs. Another indication of cellular preference or activation states. But back to SARC. So despite many of these compounds being de novo SARC family inhibitors, or at least known tyrosine kinase inhibitors, the only compound that has an effect on SARC in K562 that is comparable to the other cell lines is dasatinib, which you see here with a very tight triangle. We don't know exactly why this is, but it seems that K562 has an activation state for SARC that matches this SARC inhibitor better than others. And as you may know, dasatinib is in fact known as a SARC inhibitor with a clear niche in the treatment of leukemias testing positive for the Philadelphia chromosome bearing the fusion protein of BCR-ABL. And this happens then to be the source tumor for the K562 cell line. So it may be that dasatinib is so unique because of the good match to the SARC biology of the Philadelphia positive cell types that is not represented elsewhere. And just as a control, the protein fetch, which is frequently hit by kinase inhibitors, we compare it to the JES1 response, and as expected, there is no correlation between these two proteins, and we can't really see any clustering of compounds based on cell type, and this strengthens the previous hypothesis of activation state ratios between different SARC family kinase members that makes them respond differently to drug treatment. And with that, I hand over to my colleague, Tomas Friman, who will show you some more of these deep dives into cell biology and cell background. Thank you.
Thank you, Daniel. I'm going to talk about some other examples from our vast TA project, where we have used multiple cell lines for comparison of the same drug. First out, I'm going to talk about histone deacetylase inhibitors, or HDAC inhibitors, which we have profiled in our main cell line, HepG2, which was then a hepatocellular carcinoma, in addition to K562 cells, which are chronic myeloid leukemia cell line. But first, I will give you an introduction into HDAC and HDAC inhibitors. HDACs are enzymes capable of removing acetyl groups from histones and other proteins. The importance of this function with regards to histones is that the nature of the post-translational modifications on histones dictates accessibility to the DNA within the chromatin for the transcription machinery. The nature and sequence of histone post-translational modifications is also referred to as the histone code, and HDACs are a part of editing process for the histone code. The net effect of inhibiting HDAC function is accumulation of acetyl groups on histone tails, which eventually results in cell cycle arrest and apoptosis. This is why HDAC inhibitors has found an application within oncology. The anti-tumor effect of HDAC inhibitors is not limited to the effects of histones, but also acetylation regulation on other proteins has shown to add to the anti-tumorigenic effect. There are 4 classes of HDACs based on protein similarity, but so far only class 1 and 2 HDACs are of clinical relevance. And inhibitors against these two classes are what has been the main focus of this study. Most of these compounds are of the hydroxamate type, exemplified by vorinostat here to the right. The only exception is entinostat, which is a benzamide type, also depicted here to the right. Entinostat is also the only compound that is not annotated to have an effect on class 2 HDACs. These were investigated in the target engagement atlas format, which is 30 micromolar compound concentration, 1 hour incubation of live cells before the CETSA heat treatment. This was then performed in the two different cell lines, HEPG2 and K562. The target engagement pattern, seen here on the heat maps, are quite similar between the two cell lines with regards to HDAC proteins. In the heat map scale, blue indicate thermal stabilization and red indicate thermal destabilization. And as you can see, most of the effects tend to be on thermal stabilization. In both cell lines, the strongest stabilization effects are seen for HDAC 1 and 2 proteins, which then belong to the class 1 HDACs. Abexinostat and panobinostat has the largest effect size, whereas troxinostat and tubastatin A has the weakest effect. Also note that entinostat seemed to have a more specific effect on the class 1 HDACs, here represented by HDAC 1 and HDAC 2. In conclusion, target engagement of primary targets does not seem to differ much between the two different cell lines. Also, when looking at the CETSA effects on the global proteome, there is not so much difference between the two cell lines as well, with the exception of abexinostat, which has a large cluster of statistically significant destabilized proteins in HEPG2 cells, which are not observed in K562 cells. We went into more detail and looked at the volcano plot for abexinostat and compared HEPG2 and K562. And we can see that they have many similarities on the stabilizing side, which is to the right. They share most of the primary targets, here encircled in green, and some of their off targets or secondary targets, which are encircled in red. However, they differ on the destabilizing side, with HEPG2 cells displaying a large cluster of destabilized proteins that is not observed in K562 cells. And if we look at the correlation plot between the two cell lines, we can see this large cluster of destabilized proteins that they are clearly visible along the x-axis to the left, in contrast to proteins that are protein shifts that are common for the two cell lines, which clearly align along the correlation line. We further investigated these destabilized protein cluster and saw that it comprised 298 protein species that form a highly connected string network consisting mostly of nuclear proteins involved in RNA processing and chromatin organization. As seen on the heat map, a much larger fraction of these proteins are destabilized in red in HEPG2 compared to the K562 cells. Effects on these proteins is not that surprising with regards to the function of HDACs in regulating chromatin accessibility for transcription. What is surprising is that the effect is so much more prominent in HEPG2 cells compared to the K562 cells. This phenomenon was not observed with any of the other HDAC inhibitors in this study. This finding suggests that some feature of the abexinostat molecule elicits a much wider phenotypic response in HEPG2 cells compared to K562 cells, in spite of sharing many of the same stabilizing hits. These results highlight that there can be cell type specific responses not just to different compounds of the same class, but also towards the same individual compounds. The reason for this discrepancy between cell lines is not clear, but one can speculate about differences in baseline chromatin organization and transcription, as well as differences in pharmacokinetics of abexinostat. I will now present another example of cell line-specific differences in response to the same drug. In this spin-off project to the Target Engagement Atlas, we profiled 8 drugs indicated for the treatment of breast cancer in 5 different breast cancer cell lines, comprising representatives of 3 breast cancer subtypes. So we have BT20 cells representing the triple negative subtype. We have BT474 and SKBR3 representing the HER2 positive subtype, and finally MCF7 and T47D representing the luminal subtype. I'm going to share some of these results here. As these results are pretty fresh out of the machines, we hadn't had time to fully analyze all data yet. But we intend to present the complete study later this fall, so have a look out at Pelago Bioscience webpage for updates regarding future webinars. For that reason, I only will present data for one of the compounds today, and that's alpelisib, which is an approved inhibitor of the PI3 prime kinase, and it's developed by Novartis. The main function of the PI3 kinase in breast cancer biology is to activate the AKT protein kinase, which in turn will induce proteomogenic signals that promote growth and survival of cancer cells. The gene encoding the main PI3 kinase in breast cancer, PIK3CA, is often mutated in breast cancer, and this is also the case in the majority of cell lines in this study. Activating mutations of PIK3CA functions as oncogenic drivers for cancers. Alpelisib's function is to perturb the proteomogenic signals that stem from an active PI3 kinase. Even though PIK3CA is detected in these cell lines in the study, we cannot observe any shift, which is consistent with our previous results in other cell lines and with other PI3 kinase targeting drugs, including wortmannin. However, we observed shifts of other proteins, but the number and identity of shifted proteins varies between cell lines. So there is a clear cell line specific difference in the CETSA response to alpelisib. Most of the cell lines under investigation carries a PIK3CA mutation, where SKBR3 is the only exception that has no annotated mutation in this gene. However, the number and type of mutations in PIK3CA varies among the cell lines that carries them. BT20, for instance, has 2 mutations, of which one is annotated as an activating mutation. BT20 also displays the largest number of shifted proteins in response to alpelisib. However, the same activating mutation in PIK3CA is also present in T47D cells. But this cell line has the least number of shifted proteins in response to alpelisib. So there seems not to be a direct correlation between PIK3CA mutation status and the response to alpelisib. However, there is more of a correlation between the number of shifted proteins and the growth inhibitory effect of alpelisib, as shown here with this ATP light cell viability assay. BT20, which has the highest number of shifted proteins in response to alpelisib, also displays the strongest growth inhibitory effect in response to the same drug. BT474, which has the second most shifted proteins, also display the second largest growth inhibitory effect to alpelisib. So there seems to be some sort of correlation between the growth inhibitory effect of alpelisib in specific cell lines and the number of protein. More hits, more growth inhibition in this 24-hour growth viability assay. But more specifically, this correlation is connected to specific protein shifts regarding members of the forkhead box O, or FOXO family of transcription factors, here represented by FOXO3 in 3 of the cell lines, BT20, BT474, and MCF7. FOXO1 is also shifted in BT20 in addition to FOXO3. What is interesting here is that the FOXO family of transcription factors are downstreams or known downstream substrates of AKT, which in turn is activated by PI3 kinase, and PI3 kinase is then the target of alpelisib. The role of AKT is to inactivate the transcription factor function of FOXO proteins, which result in pro-growth and survival signals in cancer cells. In this example, we can follow the downstream effect of inhibiting PI3 kinase signaling at the third step, effect on the FOXO transcription factors. This is an important feature of intact cell CETSA, as even primary or secondary targets can't be observed to shift. The downstream biological effects can still be observed. Note that CETSA experiment only comprises an 1-hour incubation of cells together with a drug. An incubation time that is enough to see the thermal shift of FOXO in response to alpelisib, but not to assess the biological endpoint of growth inhibition. In this case, the thermal shift of FOXO family transcription factors becomes a predictor of cell growth inhibition by alpelisib observed after 24 hours. The discrepancy in protein shifts between cell lines, especially with regards to the FOXO proteins, probably reflects the activation status of the PI3 to AKT pathway, which probably then differs among the cell lines. So, in summary, we and others have in numerous examples shown that mass spec analysis of CETSA samples is a valuable tool to monitor target engagement, as well as downstream effects after short treatments with small molecular weight compounds. CETSA is a unique method in that sense that it does not require any modification of test compounds, and it can be applied to live cells as well as cell lysates and other biological fluids. TMT labeling of peptide has greatly enhanced our capacity to analyze many different compounds and conditions, as machine time has been reduced while still maintaining a good proteome coverage and quantitative precision. The quantification precision also helped us delineating biologically significant differences that are dependent on cell background. As we have shown in this presentation, the cell background matters. So with that, I would just like to say thank you for your attention, and thanks to Thermo Fisher for allowing us to have this, to be part of this webinar. We also like to acknowledge our colleagues in the lab, Lawrence Arnold, Merve Kashal, Tomas Tolvanen, Alexei Chernobrokin, and Stina Lundgren. Thank you for your attention, and here is a disclaimer. Thank you.